Hi, Nicole Mashburn again, and we're going to continue our discussion of the cell membrane, talking about transport. How do we move things across the cell membrane? So what we want to talk about is how do we move solutes, which is whatever thing is in the fluid environment. Maybe it's a sugar, maybe it's an ion, something that needs to be moved. How do we move that solute across that plasma membrane? And so just for review, you remember the plasma membrane is a bilayer with hydrophilic heads and hydrophobic tails, which basically makes a barrier from the outside, the extracellular environment and the intracellular environment, or the cytoplasm. So how can things move through this phospholipid bilayer? Well, there's several ways this can happen, and we're going to go into a lot of detail on these different types of transport. But in general, you have diffusion, which diffusion, things can just simply go through the membrane. Sometimes we have what's called facilitated diffusion, where it needs a little help. It can't go through the membrane itself. It needs a channel or a protein. Sometimes it requires energy. Maybe you need a protein that acts like a pump. So sometimes you need to pump things in or out of the cell. And sometimes you can do something called endocytosis. This is for large molecules. Large things that need to, almost like you're eating something, you're grabbing it and engulfing it or spitting it out. And we'll talk about that as well. So these are the different ways you can move things across a membrane. So let's start with the different types of transport and let's divide them based on how energy is involved. So when we think of membrane transport, we talk about passive processes and active processes. The difference is ATP. One requires energy and one doesn't. So no energy is required for ATP. All right, and I'm going to talk about different examples of that in just a second. In a passive process, the substance moves down its concentration gradient. So it goes from an area of high to low, and I'll explain that in more detail. Just hang on. Active transport, this does require energy, and this has to happen in a living cell because in order to have ATP, you have to have a cell that's alive. So living cell membranes, there's ATP involved, and this allows you to move a substance against its concentration gradient. You can take it from low to high. That requires energy. You've got to pump it. So let's talk about some passive processes, and then we'll go and talk about some active processes. So some examples of your passive processes are simple diffusion, and I'm going to give you examples of what these look like and how they work. Carrier-mediated facilitated diffusion, channel-mediated facilitated diffusion, and osmosis. Now what these two are telling you, when it says facilitated, that means there's some kind of a membrane protein involved, okay, like a channel. Osmosis is the diffusion of water. And we're going to do a separate video just about osmosis, okay? So let's talk about what is diffusion and how does it work. So what is diffusion? When you think about a beaker of water or a container of water, one thing that you need to know is molecules are always in motion. So think about water and how we talked about hydrogen bonds, and those bonds are breaking and forming all the time. And things that are in solution, they're constantly moving. The molecules are always moving. Without any energy involved. And so unless you block that movement, things will move through a solution. So in diffusion, things go from high concentration to low concentration. They move down their concentration gradient. So what do I mean by that? If I was to take a beaker of water and I put in a color disc, okay, the color is at its highest concentration in this disc, right? That's where most of the purple is. There's nothing out here in the water. Well, if I do nothing and I walk away and that color disc starts to spread out, it will diffuse. That color will start to spread out from the high concentration to the low concentration. So just walk away, and over time it will diffuse throughout the beaker until finally it reaches equilibrium where it's all equally distributed. So the concentration of the purple dye is equal all throughout the beaker. That's diffusion, going from high concentration where the purple is out to low concentration where there's no purple. That's diffusion. And this can happen, so think about even in a room things can diffuse. If you were to spray some air fragrance, it would be really concentrated where you sprayed it. It would move throughout the room or diffuse throughout the room from the high concentration to low concentration until the entire room would have an equal amount of fragrance spread throughout the room. Simple diffusion. So there's several types of diffusion. You have simple diffusion, which is just where things can move through the phospholipid bilayer without anything involved at all. They just go from high concentration to low concentration and go right through the bilayer. If it's facilitated, that means you have some kind of a protein. It can either be carrier mediated or channel mediated. So a carrier is a protein that grabs the solute and then moves it to the other side. And doesn't require energy. It still uses the concentration gradient. The concentration gradient kind of acts as the energy force that moves the solutes through the membrane. In channel mediated, the channel really just acts like a hole. It acts like a pore, and things move from high concentration to low concentration. Then the final type of diffusion is osmosis, and that's the movement of water through the membrane. And so we're going to talk about that in a separate video. So again, osmosis is a special form of diffusion. We're just talking about water movement through the membrane. Now the other type of transport is active transport, and this is the transport that requires ATP. And this is where you're going to move solute against a concentration gradient. And there's two forms of active transport. There's active transport that requires a carrier protein, and a lot of times you're going to see the word pump associated with that. And there's vesicular transport, and these are big vesicles or big pouches or big sacs, if you'll want to use that word. I'm going to show you that in just a second, to transport really large substances across the membrane. And so when I say it's moving it from high concentration to low concentration, that was diffusion. For active transport, you're going to go from low to high. So an example I use in my classroom is, let's imagine that you have a stream, water stream. And so if you're going to get on the stream, and so you're going to lay down on the stream, and the stream is moving in one direction, and you just float. You're going to float downstream with the current in the direction that the water is flowing without doing anything. Now, if you want to go back upstream, what do you have to do? You got to swim. You got to use energy. You've got to swim upstream. You've got to use some energy to do that. So active transport is when you're moving things from low concentration to high concentration. It requires energy. You have to have a pump. You have to have energy to move things against that concentration gradient. So as a review, when you move things across the membrane, there can be passive diffusion where things simply go straight through the lipid bilayer. You can use a channel, a protein, right? You can use a carrier protein. These are all moving from high concentration to low concentration, or active transport. So in this one, you can see the size of the lettering tells you the concentration of the molecule. So you can see there's a small amount of sodium here, a large amount of sodium here, a large amount of potassium here, a small amount of potassium here. And this is called the sodium-potassium pump. And it uses ATP. It breaks that ATP, breaks off, remember ATP had 3 phosphates? It breaks off one of those phosphates, which releases energy. Remember, if you break a bond, you release energy. And by doing that, that allows you to pump sodium from a small concentration out. It can pump it against its concentration gradient. It can pump sodium out. And at the same time, it can pump potassium into the cell. We're going to talk about the setup of the cell, where you expect to find more sodium and more potassium later on. But this is kind of one of the keys to how cells maintain their ion differential between the outside of the cell and the inside of the cell, is because they have something called a sodium-potassium pump that allows you to pump sodium against its concentration gradient and potassium against its concentration gradient. So remember, active transport requires energy. And if this is not review for you, and it may or may not be, depending on your background, please take some time to look in your textbook to read more about this, because this is fundamental to the understanding of cell biology, is to understand how things move across the membrane. All right, the last type of active transport I'm going to talk about today is endocytosis and exocytosis. And so remember, cyte meaning cell. So endo, bringing things into the cell, exo, taking things out of the cell. And in both of these, you're going to make a vesicle. And so a vesicle is literally a sac. And so with endocytosis, if something's outside the cell, then the plasma membrane will kind of wrap around whatever it's going to bring in and form a vesicle and then bring that into the cell. Endocytosis. With exocytosis, the opposite occurs. Whatever's in the cell will then fuse with the membrane. So you would have, so let's say this is X, and so this is going to then fuse with the membrane and then spit whatever it is out. So endocytosis brings things in, exocytosis takes things out. These are large things. They're too big to go across the channel, too big to use a carrier. So these are needed to, like a bacteria, that's huge and needs to be grabbed and brought in. So something like a macrophage, one of your immune cells, would endocytose things. We're going to talk about exocytosis when we get to the muscle unit. We talk about neurotransmitters. One of the ways we send neurotransmitters out is we exocytose them. Now there's two special words you can use when you use endocytosis: phagocytosis and penocytosis. Phago means to eat, peno means to drink. So this would be cell eating, and penocytosis would be cell drinking. So with phagocytosis, that's when you're going to get a solid particle, form a vesicle, and bring it into the cell. Penocytosis is when you invaginate and grab liquid and bring liquid into the cell, so cell drinking. And there's one more thing called receptor-mediated endocytosis, and our cell membranes will have receptors on them, and if something binds that receptor, matches it, think about a puzzle and two things come together that match. If something binds that receptor, then the plasma membrane will form that vesicle and bring it in. And we'll use that as an example later on in the semester as well. So endocytosis and exocytosis are two forms of vesicular transport. Again, take some time and look in your book if this is not new. If this is new for you, if it's not review, because we do want to make sure you understand membrane transport. That's all I got for you today. Thank you for your attention.